Thank you all for coming today. I know we've had a lot of talks on how AI is entering the landscape of arts, of finance, and also of media. On this panel, we're going to dive into how AI is diving into the landscape of something very different, which is the code of life. We're going to have discussions on how it's impacting our ability to cure biology and come up with new therapeutics, and then also how is it changing the national security landscape. Over the past months, we've had a lot of new validated AI methods come out into the open source. So on one hand, we had scientists at the Arc Institute that came up with the first AI-designed viral genome. So this was a wet lab validated bacteriophage. We also had entire new scaffolds for antibody design that were on par with what a lot of the private therapeutics companies are developing in-house. So with the picks and shovels for therapeutic design now completely open source, the playing field has completely leveled. What used to just be in the hands of private companies is now something that is now in the hands of millions of people that care to have access. For those that are bio-curious, a lot of this panel is going to dive into what does that actually mean in practice. So when you have a new paper that you see on Twitter, when it's being released, how does that actually design a new drug? Or how does that change our biodefense posture? With me on the panel are the best people that we've gathered to answer those questions. So Kenny, CTO of Latch Bio, a company that's building data infrastructure for biotechnology. Sam, co-founder of Dyno Therapeutics, who are developing viral vectors for gene delivery. And then Kathleen, CEO of a Lux company called Valthos that is building biodefense infrastructure. I'll kick off with Sam. You guys are one of the only companies that actually have a wet lab where you're testing many of these methods, particularly for developing biological products. So that's not therapeutics. Those are viral vectors that you use to deliver gene therapies. You guys are also one of the only companies that have several licensed vectors that are currently with biopharma where they're actually evaluating the use of them. So you've seen many generations of AI methods being put into practice. I would love to know your experience of actually using them. Where do you see state of the art, and then where have they worked versus where have they not worked?
Thank you, and pleasure to be here. I will start with a 20-second just intro of what Dyno is. We see ourselves as a genetic agency company, and genetic agency is defined as the ability of an individual to take action at the genetic level to improve their life, to live a healthier life. And the viral capsids that we are talking about today are an important bottleneck of this process. It's a whole ecosystem of things you want to fix to make it possible for people to take action at the genetic level. You can think of surgery as an anatomical agency and genetic agency being sort of where surgery was 200 years ago. And as you said, we had the pleasure of licensing our first AI-designed capsid to Roche, one of our pharmaceutical partners. For these capsids that we designed, these complex proteins that precisely need to target one part of the body and nowhere else so that they can be used to treat some genetic illnesses. We licensed our first capsid for neurological diseases earlier this year. And the key thing I would like to say is I'm a big user of many of the creative tools that you see today, talked about today, Runway, Suno, I'm a big fan. And actually, we just don't use it as users. We see it as what protein design could look like in a few years to come, where it's much more of a creative process and the scientist is sitting where they can use their creativity and imagination more than having to go through the mechanical process of creating designable proteins. And the trend we are seeing with these public and private tools is in the past 5 years since, say, AlphaFold came into the scene, which they deservedly say they save scientists billions of years of time. They can fold a protein in minutes that would have taken years to actually figure out in the lab. We see this trend of the floor coming up, just like the creative tools were mentioned earlier in the summit. The floor is coming up really, really fast. And for instance, internally, we see that processes that took, say, 10 scientists for the design and a year of experiments in a monkey, now we can do with one scientist and a few weeks of design, even less than a few weeks of design, and get the same performance. So that trend is really, really dropping, and the expertise and diversity of expertise that you need to be able to design some proteins, at least for the early discovery stage, has really been growing really fast in the past few years. I would say the part that is hard, and unlike, say, creative endeavors like video, in a protein, when you generate a protein, you cannot evaluate as fast as you can evaluate whether a video is correct or not. And many of the videos you generate with AI or music you generate has little problems. You can either overlook them or you can fix them later with iteration. The problem with AI for proteins is the clinical evaluation in humans takes a long time, and even that little problem will make the drug not work. So that's the barrier we have to overcome, connecting the AI tools to humans.
Thank you. So cutting down the number of people that you actually need to perform a task is the main area that you've seen impact.
Most impactful. There are many areas of impact. Yeah. I would say that's.
So that's on the molecular prediction and design side. There's also a lot of other areas where AI is entering the biosciences. Kenny, you guys have the unique vantage point of now having worked with over 100 different biotech and biopharma companies, and you actually work under the hood where you can see what their R&D pipelines look like. So I think you guys have a unique perspective on where different types of AI systems are actually coming in. What are some of the categories where you're seeing some advancements?
Yeah. There are kind of two major categories of these systems. The first is what Sam alluded to. It's these molecular prediction models. Think about these as like the outputs of the tasks are chemicals or more emergent interactions between things. So AlphaFold came onto the field and demonstrated that you can take a sequence, predict a structure several years ago. That was actually kind of the existence proof. From there. A lot of other things started to work. More recently, we had Boltz, Germinal, Chai, where it's a more complex problem. If for some arbitrary molecular target, can I produce a molecule that will bind to it? We'll probably see these just continue to move what I call upwards in the stack. Pathway modeling, cell-cell interaction, whole tissue biomarkers, so forth. There's actually no fundamental reason why it will not continue to work. In fact, like I said, the earlier problems were the more existential. The second category of things are thin scaffolding around general reasoning models. So think about these as literature distillation problems, literature search problems, experiment planning, in the early signs of agentic frameworks. So for both these categories, in practice in biotech, you have to work with the industry. So, and this is the thing that separates a cool engineering project from a product and separates you from making money and dying and not. So top of mind for us, clearly. For both these problems, the reason why you're probably not going to see a lot of adoption in the next 3 to 5 years is because to actually get people to use them, you have to understand how capital is allocated, how teams are built, and the priorities of these companies. If you take a germinal, which can generate an antibody that binds to a target in a 1 in 20 hit rate, and you compare it to hybridoma assays, which generate binding things on anywhere from 1 in 10,000 to 1 to a million, depending on the context, what are you going to do with that? So you're going to go to a biotech company, you're going to go to a team, and you're saying, oh, use this new thing. What happens to the discovery team? What happens to the decision makers with white hair at like the top that are actually going to sign off on the use of this thing? And then these effects kind of ripple through every next node in the end process of drug discovery. So if you're talking about something that's going to have an impact on the order of 6 months to a few years, you kind of have to look at what people are already doing and where they're already stuck. So from our vantage point at Latch, the big reason why lots of energy and capital has been thrown into this space is because the data generation curve. In every field of science, when data generation reaches some threshold, the way that you can build things improves a lot. And we are on a curve that exceeds the rate of Moore's law. This is coming from certain kits and machines. Within each kit and machine, scientists are currently sitting on lots of data they can't analyze. Each company has tried to spin up these jerry-rigged organizations that try to process it on their own, but they're not really successful. It leads to like many months of like back and forth between a scientist and computational. I'm skipping over lots and nuance. I'm going to have to end soon, but the point is the early signs of agentic systems that will have an impact on scientists and the rate of discovery in the next 6 months, not in the next few years, is an agent that is able to translate the natural questions a scientist already has into the concrete steps of analysis. And at Latch, we started working with Anthropic in the past month and have repurposed our entire platform to be used autonomously as tool calls with the correct context, working with the direct kit manufacturers themselves to actually finally answer these questions. I have long been very skeptical of these things, but this is actually starting to work. And this is, someone earlier asked about the mechanics. How do these affect discovery? This is perhaps a more clear idea of how those mechanics work.
And if you're not skeptical at this stage, that means, that says a lot.
Maybe.
So we've had molecular prediction, molecular design. We've now discussed also drawing analysis from the huge amounts of data sets that these biotech companies are generating. The other area that I think a lot about personally, but I think has become more pressing over the past couple of months and specifically this year, is in national security. So given that we now have an open source model and many techniques that are coming out to design entire viral genomes from scratch, the knowledge is out there. How does that actually impact, our defense posture, and how should we be thinking about that? Kathleen, I know you're also spending a lot of time thinking about this.
That's all I think about. Yes. So when we're talking about the national security side of this, the fundamental problem is asymmetry. So it's easier to make a pathogen than it is to make a cure. And that's what's underlying the entire problem we're facing. And this was true before AI. We were joking the other day, and Kenny reminded me that assembly is all you need to make any kind of virus. So we've long been worried about different kinds of bad actors using bio as an attack vector. The problem with some of these techniques we're talking about is it's really changing that level of asymmetry and not in the way that's helpful for us. So there's two big themes about why asymmetry is changing. One is much more about who is capable of making a weapon and how fast they can do it. So this is really what Sam was talking about. We've always been worried about nation state programs for this. Our DOW Chem Biodefense program specifically calls out North Korea, Russia, Iran, potentially soon China, as having active bioweapons programs. The issue is when we start thinking about the kind of uplift you could get from AI systems, we're not talking about hundreds or tens of people working in a very state-sponsored lab. We're talking about a couple people in a basement or in a dorm who could actually make something quite pathogenic quite easily. So then the other side of that is what we're probably a bit farther away from, is what do these threats actually look like? Are they completely novel? Are they adapting things from nature? Are they taking things that used to infect, maybe an animal host, now in a human? Going after agriculture is a big worry. Could it go after a monocrop? Those are the things that more capable, more accurate, more when we're talking about molecular design, could really accelerate. So when you have both of these, we have a bunch of new tools. We have the good guys and the life science companies trying to apply them to make better therapeutics and cures. We have the adversaries, and if we're being realistic, they're also going to use the same tools, and they're going to use them in ways that aren't safe. The adversaries are working on a much easier problem of making a pathogen than what we're working on. So that's the big issue at the heart of this. To not just completely leave this on a big downer, I would say the good news and how we want to try to combat this and how we want to change this landscape is using a lot of the tools that Kenny just talked about how can we get faster. And the only deterrence is updating medical countermeasures as fast as new threats can come online. So if we can update our full medical suite as soon as something emerges, it creates a great deterrence because bio just doesn't become a promising vector of attack. The way that we can do that is helping government, helping life science actually have access to this full set of AI tools that can make them faster development, more accurate, more precise. And that's hopefully what we're going to start seeing in the next couple years, faster than our adversaries.
Yeah, and I wanted to just emphasize some of the points. Of course, if you're trying to design a clinical drug, you have to be super, super precise and eliminate all the possible side effects, and that's complicated. But if you're trying to make a pathogen, you can try a bunch of things and see what works, especially if you're operating in a domain of a lone wolf or some small number of experts. And I think the example you gave from the Arc Institute, we could probably assemble a phage with bioinformatic approaches for 10 years now and make some changes to it. But right now, the bar has just really dropped to a few grad students and a few tries. And it really, that's the part that is deserves some attention to be paid. And the other side of it is obviously as precision gets really, really good, nation states are interested in precision. One of the main reasons biological weapons have not been a very big sort of a way of fighting wars is that it comes back and bites you as well. But if you can be very precise about it and only affect the sort of populations, crops, whatever strategic assets that you want, then they might become a viable tool. So it matters. But I want to say, as a person who's working in this general field, really, I am optimistic we can find ways to control this risk and, for instance, making genetic agency a possibility. We have a whole conference on this on November 11th. If any of you want to hear on the positive effects, come talk to me after. I can extend you an invite. But there's a lot of ways that we think this technology is going to be really, really valuable and important to pursue, but be responsible and diligent about what other things it enables.
And just to get a sense of how much the capabilities are, changing. If you had to have, 10 people making Evo2 bacteriophage, 5 years ago versus now, like, how do you think the number of people have changed?
Yeah, I think 10 years ago or 5 years ago, you needed like 10, 15 really highly trained bioinformaticians and virologists, etc. And now it's a few grad students who are good at AI. And then I think in 3 to 5 years, we will be looking at a person who's a hobbyist and can use it. I think that's definitely within the realm of possibility. I'm not a musician. I can make music with Suno that is comparable to an orchestra from 40, 50 years ago. I'm not as good in terms of taste, but still it's like passes as an orchestra. And I think that's the same sort of dynamics that I see in protein design.
Awesome.
Sequence design.
And then one of the things that you guys mentioned earlier was this idea of like precision. In order to actually have the machine learning make actual drugs, we need to make scaffolds that are precise. We need to bring them through clinical trials. That's a very rigorous process. In terms of where we are today, everything we discussed seems to be more on the R&D side of biotech. So I'm curious where you guys see some of those, uplifts streaming through into clinical trials. What do you see as like the biggest bottlenecks today to actually get some of these designs into human populations?
Yeah, I think that the earlier speaker mentioned, like, even if you shrink each step, it doesn't really matter because the success rate's still like 1-2%. I think that's salient. People talk a lot about Ehrum's law. So it's like the increasing cost per drug. And a lot of that is because the drug mechanism space has been picked over centuries, and a lot of the low-hanging fruit is kind of exhausted. We're also discovering that a lot of these diseases are pretty murky and very individual. So it is very convenient that this technology arise. I'm not saying this is panacea, but this technology arise that can ingest arbitrary molecular information, potentially condition on the individual molecular profile of each person, and reason about turning 5, 6, 7 knobs at once instead of one. So all that to say is I think the best way to tackle the end-to-end translational problem is getting it right from the outset by targeting individual biology in a context that will translate well, with better in silico modeling and so forth.
Yeah, I would also say genetic agency in our context is exactly about this, making it individualized and bringing all the context that's needed from clinical data, from experiments, from R&D parts of the pipeline. But R&D is, as you said, a very tiny part of the drug development pipeline. A huge amount of risk and time and money is spent on getting it to work in humans and testing it for its safety in humans. And that part is still quite a bottleneck, and AI hasn't percolated in it as much as we would hope. And there is sort of regulatory aspects that are going to be important to adapt. I think there are some signs that this is recognized and acknowledged within the government as well. But we need to be able to validate our approaches faster than we currently do to really benefit from the sort of increase of improvement of technology on the R&D side. And so that's something that comes with a lot of nuance, as it also has implications for the national security aspect of it. So I'll just leave it to you.
Oh yeah, I think the incentives there from our gov side are also that we need, if we're actually facing a threat, the speed of development of a countermeasure is so critical that like thinking through what kind of regulatory changes need to happen for that is incredibly important if we're talking about how do we change an antibody slightly and roll it out as soon as possible. So I do think like from the government perspective, the momentum is in that direction too. And there's like the national security, national competitiveness side that needs that to happen. And then hopefully also pressure from the industry on how they can rethink these things.
An example of it is recently there was a baby that was treated with CRISPR therapy, that the therapy was developed for that specific baby within a few months and executed because it was a special case. But it's a case to celebrate both innovation, the speed of execution, and the benefit that it brought to that specific child. And I think a lot of the future of the design is going to be of this kind. Drug development is going to be of this kind, where you go for that individual patient, the specific condition they're in, and you integrate all the technologies you have, AI, design, molecular technologies and computational technologies together to be able to make an impact that to date was completely unimaginable without it.
Awesome. I think we're up on time. Thank you, everybody, for listening and learning about where AI is entering R&D, clinical trials, and then also national security.